the aim of the presentation is, what the cancers are, and describe where the data is coming from. And then I'll take you through some analysis in the Biomedical Genomics Workbench, which will be focusing mainly on quality control of the sequencing data, how to do the mapping and the quantification, and then how to do the statistical analysis to compare these two cancers whilst controlling for confounding factors. And the confounding factors in this case are mainly that these studies were done completely independently of each other, so there's a very clear batch effect between them. And for the sake of keeping the number of switchovers very short, what I'm going to do is I'm going to talk about the quality control and the mapping, then do the demo of everything, and then switch back into the presentation to explain just what I've shown you in terms of the statistical analysis. And then we'll wrap up with a conclusion, and there should be hopefully about 10 minutes left after that for questions. So here's the background on the two cancers. Endometrial carcinoma is the most common gynecological malignancy in Europe and North America, and what we're going to be looking at today is the most common subtype of this. And it can have a very good prognosis if it's caught at an early state. The second cancer, hepatocellular carcinoma, it's very common worldwide, especially in China. It's not quite so common in the US, but there are a huge number of deaths that happen as a result of this. So one of them is a liver cancer, of course, and the other one is in the reproductive tract. And what my aim is today is to show you how to identify genes that are different between these two cancers. So that means that I'm interested in identifying genes that are differentially expressed in one of the cancers or in the other cancer, but I don't just want the common oncogenes that are expressed in both cancers. And I certainly don't want genes that look interesting only because they're housekeeping genes that are always present in the liver and never present in the uterus, for example. So I'm not interested in things that are just to do with the tissue type. And I'm certainly not interested in genes that behave differently because of this batch effect, just because these studies that I'm going to talk about next were done at different times by different people. And now just to break the fourth wall a little bit here, the real aim here is to show how to perform an analysis of a complicated data set. And these two data sets, they're very different. One of them's paired end reads, one of them's single end reads. And so I hope that the scientific question that I've come up with here interests you. But if not, then just bear in mind that these two cancers, the same principles apply if you have the same cancer in two different stages and wanted to compare those. Or if you wanted to ever do a correction for batch effect so that you could compare your data against some data somebody else has produced, or even some data that your own lab produced on a different sequencer or with a different postdoc in charge, for example. So there's some very general principles, which is what I really want to focus on here. So with that said, the description of the studies, that you can get the data from them. They're both publicly available in the SRA repository, and the Workbench has an SRA download tool. So if you type into these two accessions, you should be able to get to the reads and try everything I'm going to show you yourselves. But they're different in almost every respect. So one of them is paired end reads, that's the EEC study. The other one's single end reads. One of them's got quite long paired end reads, the other rather short single end reads. They've got roughly the same number of reads in them, but they're done on different machines, and they're both a tumor normal design. So there's 3 patients in each of these cancers that have been studied, and we've got some tumor tissue and adjacent normal tissue for each of these patients. So there's 6 patients in total, 6 tumor samples, 6 normal samples, 12 samples in total. And then there's some details about how the patients differ from one another. So we don't expect the patients to be completely homogeneous within a particular cancer. And of course, because I'm going to be comparing the one study results of that with the results of the other study, there's some things that it's necessary to correct for. I expect that all of the patients in the EEC study will be female, and I know that all of the ones in the HCC study are male, so if there's a difference in gene expression just to the gender of the patient, then I won't be able to tell that apart from the difference between the two cancer types, between the two study types. So I can't tell if any difference I see is because of the difference in the sequences, or difference in the gender, or difference in how the groups did their RNA extraction, or in how they stored samples, or in the environment in which the patients were living. So there's quite a lot of confounding effects here to disentangle. And the way I'm going to do it, the software I'm going to use, is the Biomedical Genomics Workbench. And it is incredibly easy to get to results through this. But what I'm going to show you today is just whole transcriptome sequencing. And the Biomedical Genomics Workbench has many more strings to its bow. You can do whole genome sequencing, whole exome sequencing. There's a lot of variant calling tools. You can do ChIP-seq data. You can do bisulfite-seq data. And then it's also got some built-in workflows that tie a lot of the inbuilt tools together to quickly get you to the insights from your data. And these workflows are often integrated with ingenuity pathway analysis or ingenuity variance analysis, so that you can go and get even deeper interpretation of your variants or your differentially expressed genes. And you're not locked down at all, so the workflows that we've got can often be modified and built upon. So they're sort of suggestions for built-in workflows. They should give very good results, but there's always the opportunity to tweak. And there's plugins that provide additional functionality. Some of these are free, and some of these are come at a small price. And the one that I'm going to be using today is a plugin called the Advanced RNA-Seq plugin, which is free for all of the Biomedical Genomics Workbench users. So with that said, the first steps that I need to go through are quality control of the sequencing data. So all 12 of the samples, whether they're paired end read or single end read, go through these two steps. And the first one you can see is a workflow that's built in called prepare raw data. And it's shown at the top left of the screen here, the workflow input of some reads. And they go through into these two tools, QC for sequencing reads, which produces a lot of reports showing how the data is looking, and you can use that to flag up if there's any concerns, if perhaps you need to resequence a sample. And then the trim sequencing tool, which can go through all of the reads and remove some ambiguous regions, can remove adapters if you supply them. And this is generally advised before you do anything else in the workbench. Once you bring in your reads, if you run the prepare raw data workflow, then what you've got afterwards should be used in all the downstream analysis. And then the second stage here is taking these trimmed reads out of this trim sequencing tool workflow and putting them into the RNA-Seq analysis tool. And what that does is it takes in the reads and the reference, and then it produces quantified expressions at the gene level with this gene expression track, at the transcript level, and then a report of how things went. And first of all, I'd like to focus on the output of this QC for sequencing reads tool, which is the first stage in this prepare raw data workflow. And this is just for one of the HCC samples. So this is the single end read samples, the reads of 58 base pairs long. And I've just picked out 3 different plots from this report. There's a lot of information in there, but these are kind of interesting to talk about. And the first one in the top left is the nucleotide contributions. So what percentage of each position in the read is a particular base? And what you can see is that from position 12 onwards, this is kind of flat. But then we've got this messy, spiky region at the start in those first 12 base pairs. And this is almost a fingerprint of RNA-Seq data. This is what we expect to see for RNA-Seq. So there's some bias in where your reads start, but otherwise they're pretty much uniformly distributed over your genome, and then you get these nice flat percentage of each base at the later stages of the read. And you can see that in the top right plot with the set of enriched 5-mers. This is a rather nice plot. It shows you if there's some real bias in your data. In this case, there isn't. You can see that the percentage coverage on the y-axis is about 0.1% to 0.7%. And what we're looking at is at each position, what is the most overrepresented 5-mer? And what percentage of the reads is that in? And you can see that basically it's about 0.4% for the most overrepresented one, which isn't much different from what you'd experience by chance. So this looks like a very good library. It doesn't have a lot of bias in it. The bottom left-hand plot shows the duplicated sequences. You can see that a lot of sequences are unique, and then there's this small little peak of sequences that are present multiple times, more than 100 times. And in DNA-Seq, if you're calling variants, this could perhaps be a source of amplification bias. Perhaps you'd be a bit worried about this. But in RNA-Seq, where you can have some short transcripts that are present at really high abundance, it's normally not something to be worried about. But I've looked at the set of duplicated sequences, which is also produced by this workflow, and I see that we've got poly A, which perhaps isn't a surprise. And then we've got a couple of other sequences, which when I blast turn out to be ferritin, which I expect to be in the liver here at high concentrations. So everything looks pretty good with this sample, and I can go through and do this for the others. So that's one of these outputs. There's also an output from the RNA-Seq analysis workflow, which is a report. And I'd like to briefly mention an interesting feature in that for paired end reads. And for the paired end reads, I need to talk about the EEC samples. And the interesting feature is when you get this paired distance distribution. So out of all of your reads, what was the distribution of the fragments? And if you do some fragment size selection, what you hope to see is that you've got just one nice peak looking a bit like this. And that's not what I see in these samples. Instead, I have a sort of long hump and then a sharp peak at about 200. And it turns out that this is what happens, this is what I guess is happening, that if you cut out the gel by hand on which you've got these fragments from, then you can sometimes get these imprecisions. And it looks like that's what's happened here. But it's the same in all of the samples, so I'm not very worried about it. I've also tried removing certain transcripts to see if this peak fades away, and it's not, so I imagine that it's just a distribution that's the same for every transcript. The second interesting thing to look at here is the paired end distances. And the way of thinking about this is that if my fragments of RNA are represented by these gray lines, then I've got some adapters ligated on the ends, and then my reads start at the ends. And so long as my fragment is longer than the length of a read, which is the case in this first and this second case, then everything that I sequence will be RNA, and everything will go fine. What you can occasionally find is that you have a fragment that's shorter than the length of the read, and the reads are 100 long in this case. And then the read will read across through the whole transcript, through the whole fragment, and into the adapter region. And then you'll get some adapter contamination at the end of your reads. So what I've done on this plot is I've marked the line where the length of the reads are, and everything to the left of this line could potentially have some adapter contamination. And typically it's about 3%, I guess, something like that. It depends on the shape of this peak, but I'm not particularly worried about losing 3% of the reads here. So I'm not going to do any adapter trimming. This all seems to have worked out for me. So with that said, now we've got to the demo stage, I'll show you how to actually run this workflow, what it looks like, and you'll see the plots being produced and the other things being produced as well. And then I'll go on to how you would run this RNA-Seq analysis, and then to the downstream analysis before coming back in to the presentation to talk about how the statistics is done and how to control for confounding effects. So here is the workbench, and here is a representative set of reads that I've got in the navigation area. This is one of the liver samples. I've got 29 million sequences with some quality scores attached. And the workflow that I'm going to run here is this one. But before I do that, for reasons that will become clear later, I'm going to set up some metadata associated with the reads. So the way I do that is I say a new metadata table, I set up a table, and I'm going to describe some properties of this sample. So I'm going to say tissue type, for example, and I'm happy with that, and I've now got a column called tissue type. And then I'm going to put in a couple of rows, and I could have some normal tissue, and I could have some tumor tissue. And then I'm going to save this table, which I can do by dragging it in to the navigation area. And I'm going to associate some data with this tumor sample. So I'm going to say these reads I'm going to log them as being tumor. And now what I can do is, when I open up this table again, this is the one I've just made, I can find associated data for this row, and here it is. And I can find it in the navigation area, and there it is. And I can run algorithms on it. And this will become useful later, because once you've got the metadata set up, you'll see that a lot of plots get decorated with the metadata about your samples. And that makes it very easy to do statistical analysis. So with that formality done, I'm going to run this workflow. There's some options for how I want to trim adapters, which I don't at this stage. I'll trim some ambiguous reads. So I'll trim reads such that they only have at most 2 ambiguous symbols. I'll trim those which have low quality, and otherwise everything's fine. Those are just the default options. And now I'm running it. And whilst I'm waiting for this to happen, I'll just talk you through how the RNA-Seq tool works. So the RNA-Seq tool is this one here. So I can find that in the toolbox, or I can use this nifty little launch tool and then just type in what I'm searching for. And it takes in a set of reads. And then I can choose to which reference I'm mapping them to. And then the default option for how the mapping is done is this, mapping to gene regions only, because that's a little bit faster. But I always prefer to map to intergenic regions as well, just in case some of the reads map outside of known gene boundaries or there's some gDNA contamination, because then when I'm doing variant calling later, I can know that those reads that really belong to an intron region or outside of the gene body aren't being accidentally mapped where they shouldn't be. These are just alignment parameters for the mapper. It can be fun to play with them, but I normally just use the default ones unless I've got a stranded protocol when I can set the strandedness here. And then there's some options for how the expression level is calculated. And the only thing I normally do is switch on the EM estimation here. And the EM estimation is just when you have ambiguously mapped reads that could belong to one transcript or another, or to one gene or another, and they could map equally well because the homology is so high. It's very good at working out which of those transcripts actually generated the read. And it's based this implementation on some very sound open source statistics. And in particular, it's extremely similar to how RSEM works, which is perhaps the leading open source mapper. And that's it. Now I can press finish, and I'll get a report out, and I'll get out all of these other things that I've been describing before. I'll get out a gene expression track and a transcript expression track, listing the expression levels in each of those two. I won't do that now because my computer will hiss away for half an hour or so, and it'll get distracting during the rest of the presentation. But whilst I've been talking about that, this first workflow to prepare the raw data, that's now done. And I can find the results. So it produced these 4 outputs. It produced the reads that have been trimmed so that only the high quality regions remain. And it's produced the reports that I was showing you before. So in particular, you can see there's a lot of different sections. And the ones that I was showing you are these nucleotide contributions. Here's this fingerprint region. The enriched 5ness, which, as I said, are only about 0.7%. That's not too worrying. And then the duplicated sequences plot. I get a supplementary report out as well, which contains a lot of details that you can parse out. And I get a trimming report showing what was trimmed. So you can see that after the trimming, I get a distribution of lengths. Before the trimming, everything's 58 long, and most things remain untrimmed, it seems. Now, had I run that RNA-Seq tool, what I would have got out is a series of reports and other things which I've got up here. So I would have got out a transcript expression track, a gene expression track, and a report for each of the samples. And just to show you the report, what I was showing you before is one of the sections in this report generated by the RNA-Seq tool. And the other things, they look a bit like this. They look like tables of expression that are linked to ensemble in this case, because I used ensemble as my reference genome. I've got my TPMs calculated. I know the length of the exons, the total exons for the genes. And I've got a lot of details about different measures of expression. So I've also got RPKMs. And the transcript expression tracks look the same, but now they're broken down by individual transcripts rather than aggregated by genes like here. So far, we've caught up with where I was in the presentation. Now I'm going to go a little bit further forward before I switch back to the presentation. And where I'm going to go forward to is that just like I set up this metadata table before, where I said tumor, normal, and told it about some reads, I've already done this for all of the samples that I'm going to be using, all 12 samples, 6 from one study, 6 from the other, that we're going to be using in the downstream analysis. And one of the things I forgot to show you before is that every time you do an analysis, all of the data generated by that analysis is appended to this to the sample it came from. So if you remember, I originally put in some series of reads and associated to them with this row in this table. And then I ran this workflow, which was over here, that generated 4 outputs. And those 4 outputs are now also associated with this row in this workflow. So there's a great deal of traceability in the system. And I've run the trimming, and I've run the RNA-Seq tool on all of these already. And so I've got a lot of different outputs. But what I'm going to do is I'm going to focus just on the gene expressions, the list of gene expressions for each sample, and I'll select them all. And then this workflow here is the final piece of bioinformatics in this presentation. But of course, then I need to explain what you're actually seeing once I've run it. So I take in all these 12 inputs, all these 12 sets of gene expressions, 1 for each sample, 6 for the liver cancer, 6 for the uterus cancer. And then we get a little bit more involved. So first of all, I have to tell the statistics what I want to test for, this differential expression. And I want to test the things that are differentially expressed between the 2 cancers. And the way that I do that is that this is all of the list of columns that's in this table. And the cancer is listed underneath the sample type column. So you can see it's written here as either normal or HCC or EEC. So I select that as the factor that I'm interested in. And then I want to control for the fact that the studies are done by different people at different times. Let's see if I've got this. So I know that the 6 samples that came from the EEC were done in one SRA project. The 6 that came from HCC were done from another project. And so I can control for the project here by controlling for the project accession. So if you see, that's this column here, and for the EEC samples it's one thing, and for the HCC it's the other. And I want to control for that confounding variable, for that batch effect. And then I choose all group pairs as what I want to see the testing due to. And that means that in sample type column, there's 3 values, normal, HCC, and EEC. All group pairs just means I want 3 outputs. I want HCC versus normal, EEC versus normal, and EEC versus HCC. So that's the testing that will be done. There's one more wizard step, and this is about uploading the results to IPA. So I put in my IPA account details here, and then I've chosen a subset of filters which will be applied before uploading genes to IPA, and a second subset of filters which is just even tighter, just which ones are going to be analyzed by IPA. And then I can run this, and we'll have a look at the results. And what you'll see is that as the workflow runs, the results are generated down here. And the first result that I get out is a principal components analysis of all the samples. So the way this works, each of the samples, of which there are 12, is a single point in this plot. And if I do it like this, then you can see a pattern emerging in the data. So you can see that the first principal component, in fact, I'll just do that. The first principal component, the greatest variation between the samples, is due to the study, whether this is done in the EEC or the HCC data. So it's these systematic effects of whether they were paired end reads or not. More likely it's about tissue, that one set of these, maybe all the green ones, are liver, and all of the purple ones aren't. But that's the most important difference between the data types. The second most important difference, that's along this second principal component, and that's separating the normal samples from the tumor samples. So what I'm interested in doing is comparing these HCC samples in green here with these EEC tumor samples in blue. But actually the difference between those two is a bit confounded by the fact that even the normals are different in these two cases. Then I can have a look at a heat map. And again, all of the metadata that's associated with these samples is present for visualization. And I can, again, choose to do it by sample type. And the heat map has just been asked to pick out the 25 most interesting genes. And this is customizable. You can also have it pick out genes that are interesting to you that you know in advance. Then you see that the liver samples, they all do one thing, and the gynecological samples will do another. And you can see some old friends here like fibrinogen down here, which you expect from the liver. And then I've got a series of statistical results out, and the one that I'm interested in is the difference between the two cancers. And you can see that for each of the genes that I have, it could equally well have been the transcripts if I'd selected those as inputs. I get out some p-values, some fold changes, and the location of them on the genome. And I get out a Venn diagram showing how all of these three different samples, three different statistical comparisons overlap with one another. So the workflow is very easy to run. Just to show how it would work, I'm going to not look at the gene-specific expression, but the transcript-specific expression, and run this workflow again. And as quickly as that, I'll now get out some results that are specific to the transcripts at the transcript level rather than the gene level. So that's what I wanted to show. And now I want to explain what it actually is that I've done, why I'm showing this, and how it's all working underneath. And this is the principal components plot that I was showing you before. It's just been recolored slightly here, but it's the same data. And the same thing that I was showing before, that the difference between the two studies is much greater than the difference between tumor and normal. That's the second principal component, tumor and normal. So that's something we have to correct for when we compare one cancer down here with the other cancer over here. And if you know about principal components, you know that actually the way that this plot looks depends very sensitively on the normalization you use. And what I've not mentioned, but what is true, is that all of these tools that I just showed you, the heat map, the principal components, the statistics, they internally do their own normalization to correct for different biases in the data. And there's two biases in these samples. The first one is that in the HCC, there's on average 30 million reads per sample. And in the EEC, there's on average 25 million paired reads per sample. And that means that if we didn't normalize for the number of reads, then we'd see that even genes that aren't actually differentially expressed would be slightly overexpressed in HCC compared to EEC. A second, more subtle kind of normalization we do is for when you have a small number of genes or transcripts that are very highly expressed. And I've tried to represent this here. It happens in the EEC data. And what you find then is that perhaps the top 100 most expressed transcripts, they make up a small fraction, a few percent. You can see sometimes that there's a small number of genes or transcripts that make up the bulk of the expression And if they experience a high fold change, well, you've only got a certain number of reads to go around. And that means that you start seeing less of everything else. And so you get a phantom effect whereby if something that's really highly expressed becomes even more highly expressed, then everything else has to become a little bit less expressed just to make the numbers balance. And you can correct for that, which we also do in all of the tools individually, in the PCA, in the heat map, and in the statistics automatically. I thought that this would be rather interesting just to give you some insight into what's going on in the statistics. I've shown you some p-values, I've shown you some fold changes. I've talked about how you can test for one factor and control for another, but it's actually very simple what's going on, not in the implementation perhaps, but in terms of understanding it. And the idea is that for a single gene, you might have a tumor which could be blue or red. You've got some state here, which is one factor. So here it's blue or red, it could be tumor normal. And then you've got some other factor, which here I've called shape, which is star, circle, and square. And these could be patients, if you like. And you've got then 6 samples and some expression in each sample for a gene. And the idea is that we choose one of these samples to be a baseline, and then we express the effect of the tumor compared to the normal with respect to that baseline, or of being one patient rather than the other with respect to that baseline. So we've got some fold changes. And it's easy in this case to sort of see, well, if I had this red rather than blue factor, then it's got a certain effect on the expression. It becomes rather more difficult if I suddenly permute these things. Now it's harder to say what the effect is of the tumor factor of going from blue to red. Is it really positive, or is it just, natural variation of gene expression between the samples? And the way that we resolve this, and the way everyone else resolves this, is you've got to assume some distribution that underlies your expression values. And that distribution is affected by all of these individual factors that contribute to separate fold change. And the distribution people most commonly use is a negative binomial one. And it's rather like a Poisson or a normal distribution. It's got 2 parameters, the mean number of counts, so the expression of the gene, and then a dispersion parameter, which measures, it's roughly equivalent to the standard deviation, how varied the distribution is. So when your dispersion, phi, goes down to 0, you get a more peaked distribution. And so what we do is we estimate simultaneously for each gene both what the size of these effects are, so whether this red or tumor effect is positive fold change or negative fold change, together with fitting a curve of this form to try and estimate the uncertainty, because maybe there's not a fold change, maybe it's just due to chance. And then we play another trick, which is that genes with similar average count, average expression, tend to have the similar dispersions, and you can use that to improve your estimates of the data. So you simultaneously estimate how wide this spread of expressions is and how great each of these effects is. And then you can ask, what's the chance that, that these effects really are as big as they seem? So you can have a fold change of 1.5, but if your uncertainty is so huge, then perhaps that's a fold change of 1.5, but it's not a really good p-value. So the results that we got to before, and I showed these only very briefly, were 3 different sets of results where we ended up making a generalized linear model of this form. That's how the statistics interpreted this request to compare EEC tumor, HCC tumor, and normal while controlling for the study. And the genes that are differentially expressed between the 2 cancers, between EEC versus HCC, they only make a small subset of the genes that are expressed in the individual cancers versus the normal. And the reason for that is quite simple. It's that, for example, in these 1,469, they might have had a small fold change in the EEC that was in the same direction as the HCC. And when I make these cutoffs that want to look at genes, I call them differentially expressed if they've got a certain fold change or a certain p-value. Perhaps I have got some genes in here that are also very nearly significant in the EEC versus normal. So they're also just generic cancer pathway genes, and they just by an accident of fate don't quite make it into this shared region here. So it's very powerful that you can control for this effect. You can directly compare the two cancers without having to worry about how your cutoffs are affecting the genes in the two lists that you're looking at. And to make that a bit clearer, an alternative to what I did here controlling for the study is I could have done some statistics where I took 6 samples for EEC and compared tumor and normal. I could have taken 6 samples for HCC and compared tumor and normal. And then I could have compared the comparisons. And then I get something that's actually very similar to just this yellow circle and the blue circle of genes that I got from this more complicated approach. But as you can see, the ones that I'm actually interested in are these pink ones, and they're only a subset of what I have here. So I'm running out of time. The very last slide I've got is to try and say, okay, so I've tried to do this comparison and only look for genes that are different between the two cancers, that aren't shared between the two cancers. How well have I done at that? And I've got 3 genes here just to show what's going on. And you can see that average expression in the 3 HCC samples, in the 3 HCC normal samples, in the 3 EEC samples, and in the 3 EEC normal samples. And the first gene is known to be present in EEC, and we detect that. It's not present in HCC, so it makes it into this list of significant genes that are different between the two cancers. GPC3, that's a biomarker for hepatocellular carcinoma. So that's significant in HCC. It's also got some effect in EEC, but despite that, it's significantly different between the two cancers. And then finally, we've got everyone's favorite tumor-associated gene, and that's significant in both of the tumors, and therefore it's not really significant when looking for things that are just different between the tumors. Also, I sent the results up to IPA, so the 788 genes that appear just in this pink region here. And I get a mixture of pathways out that are involved in either one of the tumors or the others that are perturbed in them. So you can see FXR and LXR, these are sorts of things you expect to happen in the liver. And then estrogen biosynthesis sounds like something that might not be completely unexpected as being affected in a cancer that's gynecological in nature. There's no more demo. I've moved that to the start. So to sum up, the Biomedical Genomics Workbench provides a simple interface to cutting-edge algorithms, to doing the quality control for data, to quantifying data accurately and finding out how much of each gene there is in each sample, and in terms of doing the statistics and normalization. And all of the plots flow through with metadata. And the results can be sent through to Ingenuity Pathway Analysis. When I was running those results earlier, they were automatically uploaded to Ingenuity Pathway Analysis. And if I had my email open, you would have seen that by now I'd probably have received an email telling me that the results were ready for analysis.
So I would like to thank Dr. Hill for the informative presentation on analyzing RNA-Seq data in Biomedical Genomics Workbench, and I'd also like to thank our participants today for attending. For more information about QIAGEN Bioinformatics products, please visit us online at QIAGENBioinformatics.com. And this concludes our webinar for today. And thank you, and have a great day.